Thank you all for being here. My name is Anish Agarwal. Behind me is my partner in crime, Dr. Austin Killer. We're both coming from Penn Emergency Medicine. I'm really excited for the 9th annual Lion's Den here at SAEM. So we're gonna just jump right in. We hope this to be a fun, interactive way for our junior researchers to really pitch ideas, Shark Tank style, but we call it Lion's Den, to a really tremendous panel. And thank you all for contributing your expertise. We know, so it's going to be rapid fire format. So we have 4 contestants, 8 minutes each. We're going to keep you on a clock, and then 5 minutes of hopefully constructive feedback and criticism. Austin and I have been on the contestant side before in previous years, and we're really happy to be here. The pitch here is for the contestants to pitch their ideas, get feedback, get connections, and look for sponsorship guidance as they sort of go through the process. The Lions, we know, are experienced, they're fierce, they're relentless, they're tough, and they're really giants in the field. So thank you all once again. I'll hand it over to you.
So we're going to do some quick introductions for our esteemed panelists. In selecting the panelists this year, we just were going for the most aggressive, critical, mean. We're really lucky to have. So real quick, Dr. Robert Rodriguez is professor of emergency medicine at UCSF. He's led research projects that impact our practice every day, developing the Nexus chest tool, and his recent work on COVID-19 and underserved populations has allowed him to advise Dr. Biden, President Biden, on the COVID-19 advisory panel.
That's his wife, though. He's a doctor.
There is a Dr. Biden. Dr. Karina Grudzin is vice chair for research and associate professor of emergency medicine and population health at NYU. She's a leader in palliative care and emergency medicine, and her research seeks to improve care for patients with serious illness, leading an NIH UG3, UH3 trial on palliative care interventions in the ED. Dr. Manish Shah is vice chair for research at the University of Wisconsin, and his work has just really changed the way that we deliver and think about delivering care for older adults. He's led more R01 and U01 grants than I could count and name. Maybe I can count that high, but we'll see. And he's mentored trainees around the country. Dr. Gina South is director of the Urban Health Lab at the University of Pennsylvania, and her groundbreaking research has expanded our understanding of neighborhood environments and how they impact health. Was recently awarded a U01 to study how concentrated investment in black neighborhoods in Philadelphia can address structural racism and upstream causes of health outcomes. And finally, the missing panelist. He's coming.
Don't worry.
No, he's coming. Yeah. He is.
He's going to come in and just be like.
2:15.
Yeah. Correct. Yeah. He's going to disrupt the session. Ted Melnick, associate professor of emergency medicine at Yale, and he's a formative expert in informatics and emergency medicine. He's a PI of the Embed trial, which uses decision support trial tools to improve ED initiation of buprenorphine. So, okay, with that, we are ready for our first panelist. The contestant. The contestants haven't known until today who the panelists have been, and none of our panelists know anything about the research. So, Dr. Solnick.
Oh dear. Sorry, how do I get it to start? Thank you.
Sure.
All right. Hello, everybody. My name is Rachel Solnick. I'm an assistant professor of emergency medicine at Mount Sinai in New York City, and I'm here to talk about everyone's favorite topic to talk about in public, which is sexually transmitted diseases. We're going to be talking about expedited partner therapy. So, why are we talking about this? We are, I know, coming out of continuing to be in a COVID epidemic, but we're also dealing with a less talked about epidemic, which is an epidemic of sexually transmitted infections. And what's also very upsetting about this is that this is completely avoidable. The graph on the right is showing data from the CDC talking about what share of STDs are known to be reported from emergency departments. As you can see, this number is really skyrocketing. For chlamydia and gonorrhea, it's gone up over 100%. And part of this problem is that we have a decreasing ability of public health to really assist with this. Traditionally, perhaps we have learned that there was partner management where health departments might have been able to reach out and provide notification, but they really don't do that anymore for gonorrhea and chlamydia, which are the main STDs. And as we know, this is a high-need population in the emergency department. People who come to the ED for STI complaints are likely to have higher health care access barriers. And this is also a crisis of health equity. There are stark racial and gender disparities for STIs, and those are coming out of a lot of economic marginalization as well as structural racism. So this is something that we should really be focusing on. So we actually already have a solution for this, and it's not really talked about much. It's expedited partner therapy, otherwise called EPT. This is a clinical process of treating the partners of patients who have STIs without seeing that partner. So they're an unseen partner. And the purpose is to prevent reinfection in that index patient, the patient that you treated in the emergency department. And it's actually legal or permissible now in all US states. And that's actually only pretty recently that the last states came on board with this. So it's something that we should be doing. There's actually also a strong clinical evidence base for this. Early RCTs showed that it was effective at reducing reinfection in the index patients. However, we still have an implementation gap. We don't really know how to do this very well in emergency departments. My prior research has shown that emergency departments, academic emergency departments, are not much practicing this. And there's a need to understand the contextual factors of like how to make this work in the ED, because we're not the same environment as where it was originally researched. So why should we study this? As I mentioned, we have some evidence gaps. These early RCTs I mentioned have been done in 2000s, and we live in a very different world now. Back then, they were actually doing take-home medication kits for their EPT, but now we live in a world where there's e-prescription mandates in 36 states and counting. So the ability to hand somebody a piece of paper or a medication kit is somewhat limited. So we have to figure out how this can even work, if it will work, for e-prescriptions. And there's also the silver lining of COVID is we have a lot more telehealth, and this presents a really viable option of a way to actually do even higher of a touch of expedited partner therapy, getting directly in contact with the partner. There's a higher expectation too for on-demand care that will really tie well into bringing the treatments directly to the people who need it. And then additionally, dating apps have actually changed the landscape. It's like not clear whether the relationship dynamics of patients who are treated for STIs are of the same as where they were in the early 2000s in terms of whether people are willing to bring treatments directly to their partner. So this is a perfect problem then for implementation science. And so for those of you who are not as familiar with this concept, it's really asking the question about how do healthcare systems learn as a whole system. And one analogy I had heard about early on that I really liked is this idea of like the chocolate chip cookie recipe. If you have a recipe but you like switch up some ingredient, is it still going to be as good? And this situation here is, well, we have RCTs that showed that expedited partner therapy worked in these like couple different ingredients of sites and of mechanism, and we have to see if these new approaches will actually work. And the goal is to speed translation of knowledge to practice, which is so important here because we're in the middle of a epidemic. So the proposal for the research is a effectiveness implementation trial, proposing a single-site pilot randomized type 2 hybrid trial based off of previously published work, using also the RE-AIM framework, which is looking into the reach, effectiveness, adoption, implementation, and maintenance of the implementation. And so we're going to be studying sort of two different things. One is the clinical outcome, does this actually reduce reinfection? And then also whether the different strategies we have for implementation are going to help on the feasibility outcomes for this. So in terms of the outcomes for the implementation strategies, what we're proposing is a factorial randomized design. And the reason for doing this is that factorial designs really are effective at trying to test multiple interventions on a fewer number of patients, which is, we do have like a limited number of patients that we would be dealing with. So it's a great way to test both of these things at the same time. And so you have 2 different levels for these 2 different interventions. So for one, we have a BPA, and we would potentially turn it on or off, randomized at the provider level. I've done pilot research at another institution which has shown this to be quite effective, and actually we're going to be presenting on that somewhere else in about an hour. And then also provider education, with the 2 different levels being emailing audit and feedback reports, which, as many of you are aware, has been shown to be pretty effective. And then also academic detailing, which is a higher touch approach, showing up to the ED, talking to folks, saying like, hey, did you know we can do this EPT thing, trying to make sure that it's actually happening on the ground. The outcomes that we would be looking at are looking at the REAIM framework, looking at REACH, looking of the eligible patients based off of an EMR review, how many actually had EPT ordered. The clinical outcomes we're going to be looking at are going to be 2. Let's list it at the bottom there. It's first whether the patient has repeat ED visits during which STI testing was done, and then whether they have repeat diagnosis, essentially reinfection of STIs within 10 weeks in our system. The screenshots shown on the right is actually showing that we have the tools right now to start doing e-prescribing of EPT. All you really need to do is create a dummy profile and then prescribe it through that. So creating a menu of options for both providers and clinicians to find what is the best approach for them, whether they want to do an e-prescription, a paper prescription, or something that we're already working to develop at Mount Sinai, which is a telehealth invite specific for STI care. And so why me? Why should I be doing this? So there's 2 things here. The contextual factor being New York City is a really excellent laboratory for doing this, in the sense that we have high rates of STIs. Mount Sinai is a helpful place because we have actually 6 different emergency departments, which are now within our system, that in the future, if this is helpful, if it's shown to be effective at a single site, we could then expand in the STEP Wedge trial to the rest of the sites. I personally have done a lot of both advocacy as well as literature work on ED STIs, spanning from systematic reviews, prevalence estimates, implementation science pilot trials, which involved clinical elements, as well as really trying to make this happen in terms of creating a website, rethinksticare.org, where we're going to post information such as toolkits that emergency departments across the country can use to figure out how to bring EPT to their SEs. And with that, I'm happy to take feedback and questions. Thank you.
I'll start, and I'll try to be more like Mark Cuban than Mr. Wonderful. So this sounds like a great project. A couple questions I had were, your outcomes weren't, you mentioned them, but can you elaborate a little bit further on those and
Yeah.
how they were chosen, and did you have any do you have a sample size that you think you might?
That was a great question. Yeah. So in terms of the outcomes, sorry, I sort of went pretty quickly through this. So the primary outcome is something of a more proximal measure. The early RCTs had looked at the main outcome of an STI reinfection, which I think is of most clinical interest. People still want to know what's happening to my index patient. I need to treat my patient. But however, I think it's going to be easier to see whether they're also just coming back to the emergency departments for visits related to STIs. So my prior work looking at ICD codes for STI visits has shown that to be a very hairy way to go about trying to figure out what visits are actually STIs, but luckily, because we're going to be looking at within the Sinai system, we can just check to see in any ED visits, did they have STI testing done? So that's going to be sort of the main outcome, did they come back and have an STI test? And then the second one is going to be about whether they have this repeat reinfection within a 10-week, and that's based off of the same time frame that one of the strongest RCTs on this was able to show effect in. The part of the reason of honestly doing an implementation approach is that it allows us to look at outcomes of the implementation, which is going to be a lot easier to power, honestly, just because their rates of like reinfection are around 10% or so, and so you're going to have a lot of patients to be able to nudge that level of infectivity. But with the implementation outcomes, when I had done the pilot on 50 patients at University of Michigan, we did already see an impact, a pretty strong impact on showing a randomized BPA on whether people were essentially like remembering to prescribe and offer EPT.
Well, again, I like it a lot. I would, a suggestion that I would make is, I see this single site, and you know, I would suggest going to more sites, even if it's like you know, a closely affiliated site, something like that. You're always going to have questions about generalizability. If if you do it at your single site, and you know you're the so-called experts on it already, people are going to wonder, well, you know, she's the expert already. Are your findings generalizable to other areas? That would be a suggestion I would make.
Thanks.
Dr. Salnick, great presentation. So as you know, in the ED, providers are very busy. Many people are very burnt out. And so tell us a little bit more about why you think these two provider-focused interventions, why you think providers will actually do it. And I'm also wondering if there are other staff in the ED that can be utilized as part of the intervention to actually be able to nudge providers to prescribe.
Yeah, so one element of this that I didn't unfortunately have time to discuss in this format is that we're also going to be focusing on the follow-up providers. Most academic emergency departments have some sort of like PA or nurse follow-up, and community sites have at least somebody whose like separate job is to do follow-up. And I think that in the environment in which we don't have lab confirmation prior to discharge, there is definitely an excellent argument to be made to be deferring EPT offering to the follow-up team, in which it'd be probably a little bit easier time request-wise to have that be an offer. And this can apply to the follow-up team as well. But I do think that in a small fraction of patients who are exceptionally high risk or high loss to follow-up, then those would be good patients for whom this would still be a very wonderful thing to be able to provide them. In terms of like why the BPA, so actually when we did our pilots at University of Michigan of those like 50 patients or so, we also paired it with like a mixed methods sort of a qualitative study into what were people's thoughts about getting this BPA, and we actually got like surprisingly good feedback. People said that like in general they didn't like BPAs, but because this was so specific and was only firing when it was specifically somebody who was being tested for STIs and empirically treated, that's only when they were going to get the BPA. They actually liked it, and when I asked them openly, like, what do you think would work to like help do EPT? People were like, well, maybe a BPA. So I think we've received good initial feedback that it's something that people actually might even want. And then in terms of the other elements, the emailed autumn feedback, as well as the sort of more scaled up academic detailing, this is something that from other similar interventions of trying to get to clinicians to change behavior have been like pretty effective. And the reason for like the dual approach is that not every environment is going to have the resources to provide academic detailing. So if we can get a response from just like emailed auditing feedback, like that'd be pretty great to be able to do like essentially a lower touch, lower request intervention.
You're talking one more line comment. I'll jump in. I'll jump in. It was a really nice presentation. I guess I have a couple of comments and things for you to think about, particularly given there's time. I really wasn't sure why you're doing a T2 trial as opposed to type 3. If you're arguing that RCTs have proven it's effective, then your really only question is about implementation at this point. And so I would think about that component. And you know, you just talked about the BPA, and the people at Michigan liked it. Michigan has a very strong informatics group that very tightly monitors their BPAs, et cetera. We have that in Wisconsin. I'm not convinced everywhere has that. And BPAs, I feel like now, are kind of like the 80-20 rule. When in doubt, choose 80% or 20%. When in doubt, choose BPA as your answer. And so I would really kind of think through whether that is the answer, because BPAs are getting out of control in a lot of systems.
Thanks.
All right. Next up, we have Dr. Andrew Moore from Virginia Tech. Give me 1 second. Apologies.
Andrew, you'll see raise my tone for a one-on-one.
Cool. Sorry. All right. I need my co-host here to. Oh, there we go. I figured it out. I lost the mouse, but we're good. All right, a big hand for Dr. Andrew Moore from Virginia Tech.
Thank you. So, my name is Andrew Moore. I'm an assistant professor of emergency medicine at Virginia Tech Carilion School of Medicine. It is an honor to be here today to share my research ideas with all of you. So I'm going to talk to you about syncope, every ER clinician's favorite chief complaint. Specifically, though, I want to talk about unexplained syncope, and I want to talk about the advances in wearable technology that can change the paradigm of how we deliver syncope care in ERs in America. Now, we know that it's a common chief complaint. It accounts for 1% to 2% of annual US ED visits, which equates to approximately 800,000 visits per year. On top of that, 30% of patients who present with syncope are admitted, and as you age, that risk of hospital admission increases. It's actually upwards of 50% if you're 60 years old or older. We also know that these are very, very expensive admissions. Ben Sun estimated that the annual cost of all inpatient hospital admissions is $2.4 billion. And if you extrapolate it out to the group that I'm interested in, the unexplained syncope patients, it's still $1 billion annually spent on their admissions. We also know from existing research that these admissions tend to be low yield in terms of diagnosis. Generally, about 60% of patients who are either admitted to the hospital or to an ED observation unit with an ED diagnosis of syncope and collapse will leave the hospital with a diagnosis of syncope and collapse. So we don't find the etiology oftentimes. This may be because the length of monitoring is too short, but we don't exactly know. Now, the holy grail of emergency care research with regards to syncope over the past 20 to 30 years has been the creation of clinical decision instruments named after the geographical location where they were thought up that identify low-risk patients who can go home from the ER. Now, Ben Sun bucked that trend when he came up with the appropriately named FAINT score, which is still awaiting external validation. But what none of these clinical decision instruments do is provide patient-oriented outcomes. They don't provide the patient with a diagnosis. So while all of these clinical decision instruments have been developed over the past 20 years, there's also been a tandem development and leaps and bounds ahead in wearable technology and what we can use wearable technology for. And that's where my research ideas come in. So no longer are the days of the Holter monitor recording 24 to 48 hours. We can now put on external loop recorders that record 14 to 30 days. We can now send patients home wearing mobile cardiac telemetry with cellular repeating devices and have real-time monitoring in the cloud of their telemetry. There's even single-lead iPhone EKGs, and if you're wearing Apple's newest watch right now, you're wearing an arrhythmia diagnosis tool. So we know from the SINAR flash study that patients who have syncope and palpitations who wear a 30-day event recorder are going to have a 24.5% arrhythmia diagnosis at 30 days. We also know from Cook's study that 50% of high-risk syncope patients after discharge are not going to follow up for outpatient testing. So do we have a missed opportunity here where we as ED clinicians can place a patch and obtain this data? I would argue that we do. So it's really important that we understand what research has been done with these tools thus far. The largest trial is the Patched ED trial done by Matthew Reed's group out of Scotland. It was 86 patients who were randomized following a diagnosis of unexplained syncope in the ER to wearing a 14-day external patch. Those patients had a 10.5% significant arrhythmia diagnosis relative to a 2% arrhythmia diagnosis in a comparator group. There's a historical comparator group. They also identified diagnostic findings in 3 in 4 patients. So 75% of patients that wore that patch had a diagnostic finding of some type. Now, when I was a research fellow at the only hospital accessible by ski lift in America, OHSU, I developed a care implementation pathway in which patients with unexplained syncope could be patched with a 14-day external loop recorder at the time of discharge, whether that was from the emergency department, the acute ED, or from the ED observation unit. When we did retrospective analysis of these 115 patients currently pending acceptance, we found that 13% of patients discharged from the ED observation unit actually had findings on their ELR that changed medical management. That's a pretty big number. So it's really important at this point that we understand the context and limitations of the devices that have been used thus far. And these are what are known as dumb devices. They're a single lead patch that a patient wears for anywhere from 2 weeks to 4 weeks. They then take it off and they put it in the mail. It's mailed back to the company. That company generates a report using a proprietary AI algorithm, and that report is then either emailed to a provider or uploaded to Epic. The minimum turnaround time for these is approximately 3 weeks. I would argue that with advances in technology, we can be faster. So my proposal is a randomized controlled trial comparing mobile cardiac telemetry monitoring placed at the time of ED disposition versus usual care for ED patients who have the diagnosis of unexplained syncope. Now, the way we'll approach this is that the treating ER physician will make the disposition, whether that is inpatient hospital admission, ED observation admission, or discharge home. We will not interfere with that, but we'll randomize at the time of disposition. So we'll create 3 groups that'll be randomized to either wearing a mobile cardiac patch or usual care. We're going to measure time to medical intervention in patients with unexplained syncope who are wearing the patch versus the control group, which is usual care. And we're going to measure the time to arrhythmia diagnosis in patients with unexplained syncope versus usual care. My goals for this project are to roll this out as a SAEM Foundation large project grant submitted this summer, and utilize the data that we obtain to submit an AHRQ Care Transitions R01 or an NHLBI R01, depending on who maybe I can find as my mentor. This is scalable technology that can be extrapolated out to other common ED presentations such as CHF and COPD. And it's important that we develop these algorithms and this data because we're about to undergo a fundamental change in how care is delivered in America. We have moved from 30 hospital systems having a waiver for the acute hospital at home in the past year to 250 hospitals that now have acute hospital at home waiver exceptions. We are going to be practicing in medicine in a field that we don't understand, and this is our chance as emergency medicine to own this pathway, and I think syncope is a fantastic place to start. So my ask from the Lions is that I would ask for a Lion who would be willing to be my external research mentor. My Vice Chair of Academics and Research requires all large project grant submissions to have external mentors to establish existing or expand existing emergency care networks. With that, I'm happy to take any questions, and I thank you all for your time.
So I don't know anything about syncope, but there's someone in the second row that might be a good external mentor. I'm wondering, so you said hospital at home, but this is not hospital at home. You're just talking about sending home with the device, right? So why don't, before you do this trial where you randomize people to get it or not get it, do we know why we're not doing it now? It seems like a great idea.
So we don't have the data. So if you look at all of the companies that create these devices, none of them have a use pathway that is syncope. They look at cryptogenic AFib, and there's a lot of use for that. But no one has a syncope care pathway for these remote external devices. Dr. Reed's group in Scotland and my former research at OHSU have all used the dumb devices, but no one stepped forward to use the real-time devices. I think there is probably, from a logistical and operations side of things, people will see impediments and roadblocks to what happens when this data gets recorded and all of a sudden we're getting a phone call that somebody has had a run of VTAC in the middle of the night. Well, we're in a unique position at Carilion Clinic in Virginia Tech because we actually, as the emergency department, run what's called our CTAC. It's the Carilion Throughput and Transfer Center. They're open 24/7, constantly staffed by nurses, and those nurses have a direct line to the transfer ED physician who works 24/7. So I think that's probably one of the impediments, and we have an answer to that.
I had a question about the cost of the technology itself and its performance. I feel like with a lot of research, anytime you're using some new piece of technology, the last thing you do is want to have the patient walk home with it, and that's exactly what you want to do. So
Yeah.
is this costly? What's its performance like? And to Karita's point, I wonder if there could be some additional preliminary data, although I do really like the study design that you're not going to mess with what's currently happening in the ED so that you'll then be able to show the excess arrhythmia diagnoses that are being missed.
Yeah, it's a fantastic question. And the cost data, looking at the external loop recorders, the 14-day devices that we've predominantly used, the cost is relatively low when compared to a ED observation admission. It's somewhere in the order of $300 or $400 for the device only, and then the EP interpretation and verification is another $400. So we're looking at like close to sub $1,000, at least in the system that I was in, relative our average ED observation admission was significantly more costly than that. I can't comment on the mobile cardiac outpatient telemetry device, the real-time device, because no one's ever done research with it. But we actually have a site visit with an industry sponsor who creates this device. They're coming to see us on Wednesday, and we're hoping that they'll give us the devices for free.
I guess I'll throw out two comments. So as we know, whenever we introduce technology, it gets used on a whole bunch of things it doesn't need to be used for. We spend a ton of money, it's totally worthless. I mean, it's the OBS unit, right?
Right.
We create OBS units, and so now if you complain of chest pain, regardless of your age, you end up in that, into the OBS unit. And so at some point I would suggest you talk to the gentleman in row 2 and think about it, because I'm still not convinced it's going to make as big of a difference, because you're going to need to look at your inclusion criteria, and your inclusion criteria is what's going to drive all of it. And so that's point number one. The inclusion criteria is going to be really the...
And so I didn't mention that, but our target population is 50 years or older with unexplained syncope. So we're aiming for that high-risk group. I do not care about the 18-year-old who had vasovagal syncope with a blood draw.
I would argue the unexplained syncope becomes a very broad definition either way. The other thing I'm just going to throw out there is that before you jump to the RCT and all that stuff, I think kind of maybe building a little bit of Ted's, you need to do that preliminary thing of, can you actually send this home with people? Does it actually work? Do you get the devices back? This, that, and the other thing. Because anyone's going to, before funding, is going to want to see that you can do this. You're pretty junior, unless you've done a bunch of randomized controlled clinical trials, then I think people are going to really balk at, do we give you, $5 million to $4 million to do that?
I am super junior, which is why I'm up here. The track record that we have, both looking at Dr. Reed's study from Scotland and the work that we've done at OHSU, with those devices, the return rate is approximately 85%. And the devices themselves are relatively cheap. The MCOT a little bit more expensive because it does have a cellular repeater device that goes with it, about the size of an iPhone. So, but I appreciate all of your feedback.
One more
Yeah.
comment.
I like it, virtually love it. I think it's, I'm all for novel sort of approaches to existing technology, and I can't commit to being a mentor on it, but I certainly would consider it. And I love the idea. I do agree that you have a few steps to go before that randomized controlled trial, and even before like an EMF grant, AEMF grant. But I think it's a big problem. There's clearly a big need. And even if you included the younger population, there's something to be gained by telling them, hey, you don't have a dysrhythmia. You haven't had a dysrhythmia on this report. So I think you would probably be better off including just the general population at large, because there's benefit to the negative group as well.
That's a good point. Thank you. I
Yeah.
appreciate all your time. Thank you very much.
Great. Awesome. We've scared a line away, which is even better. This is great. Thank you guys for your engagement. Next, we're going to welcome Dr. Levine from Cornell up. As long as I can pull her slides up. All right.
Thank you. Good afternoon. So I'm Debbie Levine. I'm an associate professor of clinical emergency medicine and pediatrics at Weill Cornell in New York City. And I wanted to talk to you today about salivary biomarkers and traumatic brain injury in children. So we know the problem, head trauma, very prevalent, a number of chief complaints that come into our emergency department. So traumatic brain injury, or TBI, large public health issue, accounting for millions of ED visits per year, thousands, tens of thousands of hospitalizations. And so interesting because there's obviously no objective test for TBI in children, though they're trying to come up with some testing in adults now. I think there's currently a serum biomarker that's been FDA approved in adults. So why does this matter? There's a tremendous variability in clinical signs and symptoms, and for those of you who practice pediatrics, it's even much harder for us to try to discern this, and we rely sometimes on parental report in the youngest children and certainly the nonverbal child. And neuroimaging is performed in about 40% of these kids. And with the help of PCARN and other clinical decision models that have come about, we've been able to tailor which children we decide not to neuroimage, but it's in a very low-risk patient population. And we still are subject to deciding in the intermediate risk whether we should image them. And this accounts for certainly a lot of lengthy times in the ED for observation and such, and even sedation trying to get some neuroimaging done. And then the other side of the coin is that neuroimaging is certainly not diagnostic in the minor head traumas because most of the time the neuroimaging is negative. So we're not able to objectively discern which child really has suffered a concussion or is at risk for post-concussive symptoms, which can occur in up to a third. So the goal is to identify a test that will help discriminate traumatic brain injury in children, especially the minor traumatic brain injury and those children that might be at risk for post-concussive symptoms. So we propose to look at salivary biomarkers, and this is because they have been shown to be altered in traumatic brain injury, and specifically looking at microRNA. So microRNA are non-coding fragments of RNA that inhibit protein translation. They're really abundant in the saliva. They're very stable to analyze, so they don't degrade very easily like a lot of the serum biomarkers. And it's very non-invasive. And when we're talking about children, it's really helpful not to come at them with a needle. And the studies have shown that they discriminate severe TBI in both rat models and adult studies. Studies have shown that salivary microRNAs mirror the CSF microRNAs in adults with traumatic brain injury. And then in small studies of children with minor head trauma, they have found that up to about 6 microRNAs are altered and consistently, and they've also seen an array of specific microRNAs that are altered in children that go on to have post-concussive symptoms. So we propose a prospective study design looking at children with head trauma who require head CT to kind of use a, quote, gold standard for these salivary biomarkers and compare the salivary biomarkers, these microRNAs, in children who have positive head CTs, those who have negative head CTs, and then a non-traumatic control group. And then also, as a secondary, to look at longitudinal follow-up and assessing whether or not these children go on to develop post-concussive symptoms, or have concussive symptoms at baseline using validated symptom scoring at preset intervals. So we propose this because our team is multidisciplinary. From a PEDS emergency standpoint, I'm very experienced in doing multiple prospective and multi-centered retrospective research. Our co-PI is a funded physician scientist in microRNA research with a laboratory at Penn State actually doing the microRNA analysis, and has shown multiple studies looking at TBI with salivary biomarkers and COVID-19 neurosequelae with biomarkers and autism and food allergies. And then we also have a team of neuropsychologists who have validated symptom scoring inventories in very young children from University of Montreal, and very well-versed in head trauma studies from the Canadian Head Trauma Study Research Network there. So thank you for your time.
So that was great. I really like your overall pitch. Does the saliva, do you have any sense of the time course of the appearance of the markers in saliva?
Yeah, that's a great question. So they can occur very soon after. They say up to about an hour.
Really?
And they last for about 2 weeks, and it may be even longer. So they're very quick, and it's very interesting because the benefit of using these microRNA as opposed to using these proteins that, like what they're commonly doing in the serum, like with these tests that they're doing, like the ISTAT tests that they're showing at the Abbott booth. These microRNAs are in these like exosomes, and they're just like they're kind of extruded from the cells, they're just like messengers, and you don't need actual injury to release them. So that's why they're so predominant so shortly thereafter. You're not like waiting for like cell death type of thing. So they actually appear very fast.
Can you talk about, let's say this works and you find there's this clear association, what's the long game for how to implement this in clinical practice? So if my kid falls off the monkey bars and hits his head and we come into the ED, what does that look like for my kid? And how does it change the sort of clinical decision making in the moment?
Yeah, so it's really interesting. I envision this as like our COVID PCR, right? Like our rapid COVID PCR. So it would be wonderful if we can have a turnaround of a salivary test doing a PCR with within an hour, you would get your result, because that's what we're doing, at my institution with COVID. So, and it certainly took us a long time to get there, but that would help us discern whether or not we need the CT, certainly in like a higher risk patient population, like our intermediate risk by like some of these prediction models. So if you found that maybe we may be able to look at different biosignatures, that some are more suggestive of more severe injury, so that would help discern whether or not you need the neuroimaging versus just minor. But I think really what I'm like passionate about is I really feel like as emergency physicians, we don't give enough anticipatory guidance to families about concussions and what to expect for post-concussive symptoms. So, and this will set the stage for more management strategies for being able to reduce those symptoms, because maybe if we find that this patient has this altered biosignature, they are at risk, we will be able to tell parents accordingly, really reducing activity, not going to school. There may be really some guidance that'll be very specific for those type of patients.
Yeah, just a general comment. So, I mean, I think this is very important. If no other reason, I think everyone always has a kid that hits their head and then, everyone freaks out. I think in the way you're thinking about this, I think you need to reframe this to what's your primary question initially? Because I heard about 4 different questions, some of which are going to be almost impossible to solve initially. And so when you're thinking this through, it is, okay, I first care about ICH versus no ICH. I later care about this, but that's something way downstream.
Right.
And then also, you confused me at the very end when you said these mRNAs are released even without injury. And so I would just make sure you have enough preclinical data to justify this, because it's going to have to be really, really solid before anyone's going to buy into the biomarker side.
They're released after injury. They just don't require like this cell death to release. That's what I mean, because they're extracellular.
In terms of connections, I'm working with the Track TBI group.
Yeah.
And I'm happy to connect with them, and we're doing something similar in adults.
Oh, that's great to know. Okay, yeah.
We're going to give you everyone.
I know. Got all the resources. It's great.
All right. Our last participant is Stephanie Teeple, who's an MD PhD student at the University of Pennsylvania. I want to point out that our other participants have all been faculty, and Steph is risen to the challenge of the Lions. Great. All right, everyone. Keep busy with that.
Hi, everybody. So I'm Steph. I'm getting my PhD in epidemiology, and I do want to go into EM. I'm so honored and excited to be here with you all today, and I'm hoping to share some work that I'm thinking about taking on in like the next year and a half. I'm coming to the end of my dissertation, and I'll have about another year of research time on my F-grant before I return to the clinical side of things in med school. So I think we all agree that an important part of understanding our patients' health and well-being and working towards ensuring good outcomes for them are social determinants of health. I think we all are also now realizing that structural determinants, which people often describe as sort of more upstream, higher level factors at the policy and institutional level are also important. In my work, I am broadly interested in examining health system financial policies, arrangements, and practices as a structural determinant of health and health equity. And I think there are a lot of ways that you can slice that up in particular, but I'm especially interested in sort of investments in and governance of various data products, including like prediction models, clinical decision support tools, and EHR data. And I think the ED is a really compelling and important place to study these things for a multitude of reasons. Number one, the enormous interest in EM and machine learning and artificial intelligence applications, but also, as I think I'm beginning to understand, emergency departments can serve as sort of a microcosm of larger anxieties and decision-making dynamics around health system finances. So I think there are several levels at which you can study equity, data, and health system finance. The lowest level, kind of like the individual product level, is where I focused my dissertation research, which I've spent examining potential drivers of inequitable performance for both a machine learning model for ED triage and another model for mortality risk prediction and palliative care. However, this approach is pretty zoomed in and is definitely only one piece of the puzzle. And the fact that this is really only one piece of the puzzle is really illustrated to me by a project that I actually wasn't able to complete. I was doing a qualitative study of a model that had been deployed into clinical practice, and I wasn't able to complete the study in part because there weren't enough people for me in certain roles to interview due to staffing and retention problems in the context of COVID. And those that were around were like really quite burnt out, which again just underscores the need to look at this broader context. And in that vein, I have two project ideas that I'd like to share with you. Project one, at the level of the health system, is a qualitative study of health system data scientists And project 2 is a descriptive case study of US healthcare expenditures and revenues related to data products, including things like partnerships with corporations, intellectual property, et cetera. So for the qualitative project, the main problem that I see here is there are vanishingly few studies on the people who actually build and deploy clinical decision-making models. There's a growing body of research on the people who potentially use them, clinicians, but very few on health system decision makers like data scientists who, of course, fundamentally shape the final product. The method I would use for this project would be semi-structured interviews, which are best for getting at sort of focused but in-depth contextualized meaning-making data. And the outcome would be data scientists' own understandings of and concerns for the equity implications of the things they build, and also what sorts of evaluations or oversight is already in place, as well as what could be possible in the future. And I just see this as essential baseline information and a precursor to establishing best practices for health system data governance. And I think interviewing data scientists is important because they often report to health system executives who are the ultimate decision makers for these things, but are hard to access as research subjects. So for this project, I already have background in qualitative data collection myself. I have a qualitative mentor picked out who will sort of help guide me through the analysis, presentation, and writing of this kind of paper. But what I could really use for this project is access, context of data scientists at other institutions. I have a few contacts myself, but I think it would be really helpful to compare and contrast maybe one or two additional institutions in this project. And then for project number 2, the main problem that I see here is we don't even know at a descriptive level how much and in what ways healthcare systems are spending and making on data products. There have been sort of scattered examples in the news of different healthcare systems, for example, like partnering with tech companies to share data or spinning up their own database startups or patenting models, but there's been no comprehensive review. So the methods I would employ here could be a couple things. First, just a review of sort of relevant industry publications. I'm also interested in doing a linked patent database literature search, and my idea there is to scrape authors' names from publications about artificial intelligence applications and then cross-reference them with a patent database search. And I think it could also potentially be helpful to triangulate these findings with some key informant interviews. And then the outcome would be if and how money is flowing in a given domain. And I think an important way to make this project more tractable and impactful would be to limit it to a specific clinical problem where there's been a lot of action, like sepsis or readmission. So in order to do this project, I think it would be helpful to have some help with the patent databases, which I have been warned are particularly opaque. Specifically, it'd be nice to speak with someone who has cleaned and used this data before. And then also, I think key for this project, it would be important to have mentorship around the framing and writing of this piece and its publication. I think we're also supposed to talk briefly about ourselves and why we're the right person to address this problem. I guess I do just want to give the caveat, I don't think anything to do with equity or science really is totally a one-person job, but I do think I'm fairly uniquely positioned to bring an important team of people together. My PhD research is in social epidemiology, and I've spent a lot of time becoming familiar with the work in the social sciences. And social scientists have been doing important work in the equity arena for a long time that I think academic medicine is kind of only recently waking up to. With my dual degree, I'm going to have direct clinical experience, up close and personal with how health systems operate, and I'm also deeply personally invested in translating my research into material change via policy, by engaging with the public, still learning the best strategies for doing that last piece, but it is a top priority for me over the next several years. So in summary, thank you so much for your time. I hope you'll consider investing in these projects and my growth in this area more broadly. Understanding structural determinants is critically important, and I think financial policy and practice are an often kind of invisible piece of that absolutely impacts healthcare delivery, health employee well-being, patient outcomes, the health and well-being of surrounding communities, and more. So thank you so much for your time. I'm really looking forward to your feedback.
I can jump in and start.
Go ahead.
Or,
No, go ahead.
So this was actually absolutely wonderful, and it's something I could definitely buy into. We have a number of things in this area that are going on. I think the 3 comments I want to quickly make. One is, I'm impressed that if someone getting a PhD in epidemiology is allowed to do qualitative research. The epidemiologists have become enlightened since at least the time I had my last doctoral student in epidemiology. The second comment I was going to say is that this is absolutely critical because all the AI stuff we're going to do is going to basically create a new structure to further racism. And it's not something everyone gets. It's not something that's gotten in the private sector quite as well. Which leads up to the third thing. And so we've got a bunch of work going on this. We have a new Center for Health Disparities Research that we're leveraging off the NIH mechanistic framework, where we are looking at some of the policy stuff, and I'm one of the assistant directors of it. And this falls very much in that rubric, and so that's really the area that you can leverage it through, you can push it through, and I think it would get a lot of traction.
Amazing. Thank you so much. Would love to talk further at some point.
I just had a question about the first project. I was a little confused. So the data scientists, explain what you mean by a data scientist and where their leverage is in this whole structural determinant.
Totally.
Because I think I have people for you, but I want to make sure
and that's
send you the right people.
Yeah, that's a great question, and I think data scientist is kind of a sort of nebulous occupational descriptor. I'm thinking of anyone who works for the health system, and actually conceivably really anybody sort of on like the health system IT side. Perhaps the more sort of like quantitative implementation.
Okay, that's what I thought.
Touching people.
Yeah. Okay, great.